Good morning, my dear student friends. I am Vijay, Assistant Professor, Second of College of Commerce, Tirupati District, Tamil Nadu, India. I welcome you all to the Vijay's classroom of biology. So, in this lecture, I am going to give you an overview about history, developments, branches, and scope of microbiology. Before getting into brief history and scope of microbiology, one has to know about the actual definition of microbiology. Microbiology is a study of microorganisms. Microorganisms are nothing but the invisible living creatures that are non-naked to your eyes. So the subject deals with viruses, bacteria, algae, fungi, and protozoa. The importance of microbiology, microorganisms can even be overemphasized in recent days as they are necessary for the production of bread, cheese, beer, wine, antibiotics, vaccines, vitamins, enzymes, and many other products. So, in recent days, the research work related to modern biotechnology rests upon a microbiological foundation. Microorganisms are everywhere. They are almost everywhere and invisible to our eyes, as if we feel the presence of God. So, almost every natural surface is colonized by microbes, like from human body to ocean. Some microorganisms can live in hot springs, whereas many other microorganisms can live on frozen sea ice surface. In simple terms, I mean the icebergs. Most microorganisms are harmless to humans. We usually swallow millions of microbes every day, causing no ill effects. In fact, we are even dependent on these microorganisms to help digest our food. Microbes also keep the biosphere running by carrying out essential functions such as decomposition of dead animals and plants. They make possible the cycles of carbon, oxygen, nitrogen, and sulfur to take place in terrestrial and aquatic ecosystems. Microorganisms have also harmed human beings and disrupted the society over the millennia. So, in line with that, microorganisms can cause several diseases in humans, animals, plants, and are involved in food spoilage even. Infectious diseases have played a very important role in the human history. So, for example, the decline of the Roman Empire was due to Antonine plague and the conquest of the New World happened. And today we have the COVID pandemic as an evidence of the infectious diseases that are playing a major role in shaping the human history. We had a Great Plague, which had reduced the population of Western Europe by 25%, and smallpox and other infectious diseases were introduced by European explorers to America in 1500s, and that was responsible for the destroying native American populations. So, until late 1800s, no one had proved that the infectious diseases were caused by specific microorganisms. This is because the microscopes were not well explored during those days as a device to study microorganisms. Invisible creatures were thought to exist long back before they were observed using a microscope. So that's the actual fact. But Antony von Leeuwenhoek was the first person who invented the first microscope that can magnify 50 to 300x. And she was the first person to accurately observe and describe about microorganisms. As a cloth merchant, during his leisure time, he started observing ponds and small insects using glass pieces which are arranged in stacks. So he started adjusting the distance between the glass pieces and he was able to magnify small objects. And with that sense, he started developing a small microscope which could amplify the image to 300 times. And those days, from the earliest times, people believed that living organisms could develop from non-living or decomposing matter. So this particular theory is called spontaneous generation theory, which was later disproved by Francesco Redi, John Needham, and Lazarus Spelunzani, three important scientists who actually challenged the spontaneous generation theory. And Louis Pasteur was the hero there. He could actually settle the conflict once for all with his swan neck flask experiment. In his swan neck flask experiment, he actually boiled the soup. He boiled the non-veg soup, and then he actually heated the neck of the flask to blew them out into a swan neck. So in this swan neck shaped mouth, air can enter along with its microorganism. But in the first bend, microorganisms get settled, only the pure air passes through and it reaches the nutrients. So, this particular flask was stable for several years without any microbial spoilage. And when he started bending it, it was actually the boiled soup was actually susceptible for microbial contamination, and it was spoiled within a few hours. Then he started bending it and then resuming it back to its original position. So it means microbes are everywhere. They are also in air, and once when they enter into nutrient medium, they multiply, and life comes from life. In a boiled material, you may not expect life to arise. Life comes from the pre-existing life form. So this is what is the actual theory of biogenesis, which is after the which came into existence after the disproval of the spontaneous generation theory. And role of microorganisms in disease. So in these aspects, we have Bassi. This person showed that the silkworm disease was caused by a fungus. And Berkeley and Pasteur showed that microorganisms caused diseases. And Joseph Lister was the first person to develop an antiseptic surgery, and he developed the system for a sterile surgery method. And Robert Koch is the father of bacteriology and have established a relationship between Bacillus anthracis and the disease anthrax. He could isolate the Bacillus species from tuberculosis infection and he could cultivate. And he was the person to propose then germ theory of diseases. Diseases are caused by living microorganisms which are invisible to you, right? Germs, they were referred to as germs in those days. And Charles Chamberlain was the first person to discover viruses as well as its role in causing diseases. He was the first person to use autoclave for sterilization technique as a sterilization method. Robert Koch was the person to postulate the role of microorganism in causing diseases. So the postulate is referred to as Koch postulate. So according to the postulates, there are four important points to remember Microorganisms must be present in every case of the disease, but absent from healthy individuals. The suspected microorganism must be isolated and grown in pure culture. Disease must result when the isolated microorganism is inoculated into a healthy host. The same pathogenic microorganism can be re-isolated from the diseased host. Isolation of microorganism, which is a useful technique in microbiology, and we have a brief history for this. During Koch postulates studies, it became necessary to isolate the suspected bacterial pathogens so as to prove that these are the actual responsible organism causing diseases. So, Koch started culturing bacteria on sterile, cut and boiled potato slices, which was not satisfactory to maintain a pure culture. Then, because when you want to actually observe the organisms, it should appear like non-overlapping individual uh, colonies. In regular liquid medium, we cannot observe as it is a single separate colony because growth is actually measured in terms of turbidity alone, irrespective of the organism. I started solidifying the regular liquid medium by adding gelatin. However, gelatin melted at a temperature about 28 degrees Celsius. So this is the room temperature or incubation condition where you cannot allow the solidification of the medium so as to visualize the colonies. Then Fanny Elsenius suggested the use of an agar. Agar actually melts at 100 degrees C and solidifies at 50 degrees C itself. So he started using agar as a solidifying agent. And Richard Petrie was the first person to develop a Petri dish, a container that can hold the solid culture medium. It's a container that has a plate as well as a lid. So in this Petri dish, we can pour the medium that is actually going to solidify into a solid matrix on which we can culture microorganisms as a single separate pure bacterial colonies or fungal colonies. And during the Louis Pasteur's time, he was able to develop vaccine for chickenpox, anthrax. After the pure culture has developed, these things could be possible. He started developing chickenpox vaccines, anthrax, as well as rabies. He was the first person to demonstrate the fermentation process. The fermentation processes were due to the activities of specific yeast and bacteria. Louis Pasteur developed a pasteurization to preserve wine during the storage. So it was very important during those days in wine making as well as beer making industries. As these industries met severe losses during the storage due to the spoilage. So he developed a method called pasteurization. Using that, we can eliminate the spoilage as well as pathogenic microorganisms from breweries. Fermentative microorganisms were generally anaerobic and could live only in the absence of oxygen. He was the first person to discover that they can live only in the absence of oxygen, and they were actually responsible for fermentation process. Because during those days, people believed that the production of alcohol is because of the decomposition of organic molecules, the natural decomposition process. He was the first person to observe microorganisms as well as their role in fermentation process to yield products. There are also other developments by other microbiologists like Vino Gradisky. He made many contributions to soil microbiology. He was the first person to discover that bacteria could oxidize iron, sulfur, and ammonia to obtain energy. actually isolated anaerobic nitrogen fixing bacteria and studied the decomposition of cellulose. And together with we have Bajering, who developed an enrichment culture technique, and he suggested the use of selective media to selectively enrich the growth of specific microorganisms. During the early 40s, microbiology established a closer relationship with genetics and biochemistry. So during those days, microorganisms are extremely useful as an experimental model system to study genetics as well as the biochemical processes and biochemical pathways. So, for example, study of relationship between genes and enzyme. So, one gene and one enzyme hypothesis was studied, and the evidence that the DNA is the genetic material was also studied using microorganisms. Recently, microbiology been a major contributor to the rise of even molecular biology disciplines. So, studies on genetic code, mechanisms of DNA, RNA, and protein synthesis, regulation of gene expression, as well as the control of enzyme activity was studied. These ones were studied using the microorganisms as a model system. And the rise of recombinant DNA technology and genetic engineering form, forms the basis of the modern biotechnology, and both of these ones involve microorganisms as the model system. So, in broad sense, the microbial world can be categorized into two major domains. Actually, three major domains. However, for your understanding, just based on the simple cell structure, we can define into two. One is prokaryotes, which are relatively simple in morphology and lack true nuclear membrane. So they lack true membrane delimited nucleus. Whereas in eukaryotes, you find true membrane enclosed nucleus. Here, the morphology is more complex in eukaryotes. So these are the two important morphology that divides the microbial kingdom into two. And one more third kingdom is about archaea, which is somehow related to prokaryote in its nuclear structure. However, the membrane architecture varies between prokaryotes and then archaea. So these archaea are having complex morphology with stable membrane, stable lipid membrane. During those days, people believed five kingdom concept. It includes the Monera, first kingdom as the Monera, having the prokaryotes, which are nothing but the organisms with simple morphology and lack a true membrane delimited nucleus. And the second kingdom is about Protista, which includes unicellular or colonial eukaryotic cells lacking true tissues, includes algae, protozoans, and simpler fungi. And thirdly, we have the kingdom fungi, which includes eukaryotic cell forms, includes molds, yeasts, and other mushrooms. Fourth kingdom was about plantae, which includes multicellular photosynthetic organisms. Fifth kingdom was animalia, that includes non-photosynthetic metazoans, which are multicellular in nature. In the title scope of microbiology, we should know about different disciplines of microbiology as well as the scope to study it for the um, human use. So microbiology has an impact on medicine, agriculture, food science, ecology, genetics, biochemistry, immunology, and many other disciplines. Many microbiologists are primarily interested in the biology of microorganisms, while others focus on specific groups. Say, for example, virologists study viruses, bacteriologists study bacteria, biology of bacteria, phycologists study the biology of algae, and mycologists study the biology of fungi, and protozoologists do study the biology of protozoans, which are unicellular protists. Under the case of different branches of microbiology, as these are the people who study different group of microorganisms. So based on the application of these microorganisms, they are called with the different terms, and we have multiple branches too. So we call medical microbiologists who actually deals with the living organisms, microorganisms that cause diseases to humans and animals. They generally identify and plan some of the measures to eliminate these agents from the biological system or from the host, causing infectious diseases. And there are immunologists who work on the discipline immunology, which is nothing but the study of immune system that protects the body from parasitic agents. And we have agricultural microbiologists who study certain agriculturally important microorganisms, and they work on how to combat plant diseases that are actually caused by the infectious plant microorganisms as well as pests by utilizing the agriculturally important microorganisms so as to protect several food crops and cash crops. Food and dairy microbiology are actually two different, two related but somehow distinct disciplines. Here, both of these microbiologists work to prevent microbial spoilage of food and dairy products as well as certain transmission of foodborne illnesses. So, for example, salmonellosis caused by Salmonella enterica. And these food and dairy microbiologists also make use of some useful microorganisms to prepare food products such as cheese, yogurt, pickles, ghee, many other products related to. And we have industrial microbiologists. They use microorganisms to make products such as antibiotics, vaccines, steroids, alcohols, and other solvents, vitamins, amino acids, and useful enzymes. We have several microbiologists who work on microbial biotechnology. They primarily work on genetically engineered microorganisms to prepare hormones, antibiotics, vaccines, and other products for human use. We have virologists too. They always work on the living and non-living characteristics of viruses. Viruses are generally acellular, and they are considered neither prokaryote nor eukaryotes. They are acellular. They are always ultramicroscopic intracellular obligate parasites. So these virologists always work on viral infection as well as the biology of these viruses, life cycle of these viruses, and the stages of infection. And they do develop vaccines or certain antivirals to protect the infection to plants as well as animals and humans. So, with this brief intro about the history, developments, scope, and branches of microbiology, my lecture comes to an end. Thank you.